Dear students, I hope you all will be fine. Here I am, Mohammad Tajamal Khan, lecturer in botany, University of Education, Lahore. Today we will discuss a topic from one of the core subjects of botany, that is plant systematic anatomy and development. The course code of this subject is BOTN 1112. This is lecture number 27. In this lecture, we discuss the vascular cambium under the content mentioned below. Firstly, we discuss simply a vascular cambium. Then we reveal the location of vascular cambium within the plant. Then we observe the vascular cambium formation in stem and root. Then we discuss the types of cell. After that, we discuss fusiform initials and reinitials, and in the end, we briefly discuss the vascular cambium activity. Vascular cambium is a thin cylindrical layer that is present between the primary phloem and primary xylem. The origin of this vascular cambium is lateral meristem cells. This vascular cambium is associated with the secondary growth of plant, as these cells divide and multiply and increase the girth of plant and play a very important role in secondary growth. Xylem, that is derived from vascular cambium, basically transports water and nutrient through the plant, and it grows inward from the vascular cambium. And the phloem, the vascular tissues that transport sugar and other large molecules, grows outward from the vascular cambium. When the vascular cambium cell divide, they create secondary xylem and phloem. So here in this slide that indicates the schematic origin of different tissues in plants. So here, first of all, here we have the stem, and here we have the root. So the tips of stem possess apical meristems, and the tip of root possess the apical meristem here. So after this, we have three types of primary meristems. One is protoderm that produce epidermis. Then we have a ground meristem that produce two types of cells or tissues. One is pith, the other one is cortex. And then we have a procambium that play a role in the formation of primary xylem and primary phloem. Similarly, in root, protoderm play a role in the production of epidermis. This is the primary tissue. Then the ground meristem that produce cortex, and then we have a procambium in root that produce a vascular cylinder, and these vascular further produce pericycle, then primary xylem and primary phloem. But here our focus is vascular cambium in stem and in root. So in stem, the vascular cambium is produced by lateral meristems. So these all structure are produced by the primary or by the apical meristem. After this, here we have the role of lateral meristem. This lateral meristem produce two types of cambium. One is vascular cambium in stem, and the other one is vascular cambium in root. In addition to this, it also produce cork cambium that produce bark of plant or trees. So the vascular cambium, after division, as I already told in my previous lectures, that this vascular cambium play a very vital role in secondary growth by the production of secondary xylem and secondary phloem. As far as we have a concern with the location of vascular cambium, this vascular cambium is present between the phloem and xylem. So here we have the cells of vascular cambium that is present between xylem and phloem. So this vascular cambium play a very important role in secondary growth. It means it increases the girth of plant. So here we have another image that clearly indicates the presence of vascular cambium between xylem and phloem. So here in the inner side we have the cells of xylem. These are the cells of xylem, and towards the outer side or periphery we have the cells of the phloem. These are the cells of phloem. In between xylem and phloem we have the cells of vascular cambium. Furthermore, the location of vascular cambium is different in dicot and monocots. In dicot stem and root, it is mainly located between the xylem and the phloem in the stem and the root of vascular plant, and it is the source of both the secondary xylem growth inwards towards the pith and the secondary phloem growth outward due to this vascular cambium. This slide gives further elaboration regarding the location of vascular cambium. So in this slide it is clearly observed that the vascular cambium, this vascular cambium is present here. So this is the location of the vascular cambium. So this vascular cambium, outside this vascular cambium, phloem is produced. So here we have the phloem, and these are the cells of phloem. And inside this vascular cambium we have the xylem. But this vascular cambium divide because this vascular cambium consists of meristematic cells, so they have the capability to divide. So after division, this vascular cambium produce secondary phloem and secondary xylem. So by the production of secondary phloem and secondary xylem, it play a role in the secondary growth of plant. So actually vascular cambium have role in secondary growth by the production of secondary phloem and secondary xylem. As far as concerned with the location of vascular cambium in the monocot stem, here we have the example of corn and palms and bamboo. In monocot plants, they don't have a vascular cambium and do not exhibit secondary growth by the production of concentric annual rings. So in monocot, they cannot increase in girth by adding lateral layers of cells as in case of conifers and woody dicots. So here we have the comparison of monocot and dicot stem. First of all, as you observed in this slide, that the cortex is present and it is composed of simple permanent tissues like collenchyma and parenchyma. But in monocot stem, ground tissues are not differentiated. That is clearly indicated in this slide. That is not differentiated into cortex and pit. So ground tissues are majorly composed of parenchyma cells in monocot stem. In addition to this, in dicot stem, endodermis is present, as clearly indicated here. It is present in dicot stem, while it is absent in monocot stem. As you observe, the vascular bundles are arranged in rings. Here you observe these vascular bundles are arranged in rings form, while in monocot stem the vascular bundles are smaller, and the smaller vascular bundles are present towards the periphery and the larger towards the center. In addition to this, the vascular bundles in dicot stem are open, while in monocot stem the vascular bundles are closed. The clear indication, the clear differentiation between monocot and dicot stem is the presence of cambium. In dicot stem, the cambium is present, while in monocot stem it is totally absent. As far as other differentiation is concerned, in dicot stem, here we have the pericycle. So pericycle are present in dicot stem, but in monocot is totally absent. In dicot stem, the xylem vessels are arranged in radial rows. Here, these are the radial rows. These are the radial rows. While in monocot stem, the xylem is V-shaped, as clearly indicated here. This is the V-shaped structure. This one is V-shaped. Furthermore, in dicot stem, medullary rays are present in between the vascular bundles, and in monocot, the medullary rays, these are the medullary rays that is present between vascular bundles, while in monocot, medullary rays are totally absent. So, as the vascular cambium play an important role in the secondary growth, so as far as concerned with the secondary growth in dicot's stem begins when the vascular cambium cell arises from a basic meristematic cell, that is procambium, or that is also called as a residual procambium. So these residual procambium cells present between the primary xylem and phloem that ultimately convert itself into a vascular cambium, and this vascular cambium is further these vascular cambium cells are existing cells that becomes meristematic at different times under the influence of different factors like hormone, and eventually they form a complete cylinders, and this cylinder runs throughout the middle of each vascular bundle to produce secondary xylem and phloem. So in this slide we observe the vascular cambium formation in root. In root, the arrangement of primary xylem and phloem, as indicated in this slide, prevents the vascular cambium from initially forming in a circular pattern or configuration. But as within a year or so, different rates of cell division in the vascular cambium result in the formation of a cylinder. So after this, it produces secondary xylem and secondary phloem due to the activity of vascular cambium. So our secondary xylem expands the plant's capacity to carry water and minerals up from the roots and add structural support. In addition to this, the secondary phloem increases the transport of food from the leaves. As the stem or root grows in thickness, the mature primary xylem and phloem tissues are pushed farther apart as indicated in this slide. As you know, the vascular cambium is a very thin cylindrical layer that is present between the primary xylem and primary phloem. So basically, the vascular cambium consists of meristematic cells. So there are two types of meristematic cells that combine to form vascular cambium. One is, we call it as fusiform initials, and the other one we call it as ray initials. So the fusiform initial, these are vertically elongated meristematic cells, while the ray initial, these are horizontally elongated meristematic cells. So the fusiform initial cells are elongated cells with taper ends. They are very long cells. The longitudinally oriented elements in an organ, such as you study in previous lectures, like trachea elements, fibers, xylem and phloem parenchyma and sieve elements, develop from the fusiform initial. On the other hand, the ray initials are very much smaller cells, as indicated in this slide. They are almost isodiametric, and the cells of vascular rays, which are oriented horizontally in the organ or in plant, develop from ray initials. So here we have the features of fusiform initial. These are spindle-like, elongated in shape. So it appears like spindle-shaped cells. So they are arranged in a vertical row, arise within the vascular bundles and produce new vascular tissues that play a role in secondary growth. So a xylem is present inside these fusiform initial, and phloem is present outside the fusiform initial. And these fusiform initial divide so that one daughter cell become either xylem or it may be a phloem. A fusiform initial cell can divide to produce two fusiform initials that remain within the vascular cambium or become the part of that vascular cambium. Fusiform initials and conducting cells are aligned lengthwise parallel to the surface of stem or root. Here we have the features of ray initials. These cells are horizontally arranged in vascular cambium, and they help in producing phloem rays and xylem rays. So here I elaborate the two terms phloem rays and xylem rays here. Mostly when the ray initial divide, it produces cells or produce cells that transform into xylem and phloem. But in some time or in some cases, certain cells don't transform into xylem and phloem and remains as such. These cells cut out by the cambium and move towards the periphery where the phloem is present, and these cells act as a phloem parenchyma or phloem rays, while those cells that are present inside this vascular cambium or towards the pith, those cells convert itself into a xylem parenchyma cells or xylem rays. So these rays are also known as vascular rays. So these ray initials arise between the vascular bundles and are often cubical in shape, and they produce cubical parenchyma cells. These cells serve mainly for storage because these are parenchyma cells, so they have a role in storage and also for sideway transport. Ray initial division leads to the formation of daughter cells and parenchyma cells. In addition to these initials are common in dicots, where they are often more than one cell wide, or their width is one cell more than other cells. So these two initial, the fusiform initial or ray initial, undergo two types of division. One is called periclinal and the other one is called anticlinal division. So when periclinal division occur, it adds to the length of stem or root, while in anticlinal division it increases the diameter of vascular cambium. As you know, both the fusiform initial and ray initial are the meristematic cells of vascular cambium. So vascular cambium consists of these two meristematic cells. So when these two cells divide periclinally or anticlinally, they increase the length as well as the diameter of the vascular cambium. So for better understanding, one can say that the periclinal cell division are the ones that occur parallel to the tissue or the organ surface. So as a result, we get rows of cells stacked one over the other. Like in this case, so we got rows of cell. This is one and this is the second row, stacked one over the other. The anticlinal division are perpendicular to the adjacent layer of cells. This one. So that you one get columns of cell adjacent to one another. So here we got column. These are the columns. This one is a column and this one is a column. This is also an anticlinal division, perpendicular and to the adjacent cell. Then we obtain columns here. In simple word, the anticlinal division add more thickness and periclinal division add length. Periclinal cell division occur more in plants than other living organisms. So simply you can say that anticlinal division is at right angles to the surface. So due to this, the anticlinal wall of a cell is arranged perpendicular to the surface of the plant body. This anticlinal division leads to the formation of anticlinal walls between daughter cells. And such type of division allows the tissue to increase its circumferences, thus increasing the girth of the organ. While the periclinal is parallel to the surface, and such a division result in an increase in girth of organ, thus adding length. In cylindrical organs such as stem and roots, the term tangential may also be used in place of periclinal division. So here we clearly indicate the activity, vascular cambium activity here. So here we have two types of cell that exhibit by vascular cambium. One is fusiform initial, the other one is every initial, as clearly indicated in this slide. So after the division in these cells, fusiform initial cell and reinitial cell, it produce secondary xylem and secondary phloem. And due to this activity, the activity of the vascular cambium is not uniform throughout the year. This cambium is most active during the spring season. So in this season, it produces wide and loosely dispersed tracheal elements in xylem that ultimately produce a wood that is called as spring wood. The activity of cambium slows down during the winter, or it may be inactive in winter. So during this season, it produces narrow and closely placed tracheal elements, and that ultimately produce a wood that is called as autumn wood or winter wood. This difference of spring wood and autumn wood produce annual rings, and annual rings can be seen in the transverse section of wood, and these rings help in the determination of age of trees. All about this lecture. Thank you.